Microbes, Metabolism and Autoimmunity. Ramnik Xavier, professor at the Harvard Medical School and member of several research institutes at the MIT and Harvard, provided in this talk an overview of the gut microbiome in early life and some connections to human health at the 14th Nestlé International Nutrition Symposium. While the incidence of various infectious diseases continues to decrease worldwide, the incidence of various immune disorders, such as allergy, type 1 diabetes, Crohn's disease, or other inflammatory bowel diseases, or IBD, is increasing and represents a global burden. These disorders have an uncontrolled inflammation in common. This increase correlates with the socio-economic changes, the absence or presence of hygiene, which includes the microbial exposure in early life, the rapidly changing diet, and lifestyle. Still, the question is, how is our gut microbiome and autoimmune disease linked together? The composition and function of the gut microbiome are profoundly impacted by the changing environmental pressure of modern life. This can lead to breaks in immune tolerance and to a broken circuit of inflammation in genetically susceptible individuals. In the first part of his talk, Professor Ramnik summarizes what is known about gut microbiome development in early life. The intestinal microbiome orchestrates the mucosal T-cell response early in life. A critical window of antigen sampling exists early in life, during which our microbiome and microbial exposure affects the immune development. How we sense and react to signals from the diet, its microbial products, and our microbiome has an impact on our susceptibility to interruptions in immune tolerance and to damaging inflammatory responses. In fact, our understanding of this complex sensing mechanism is little, and more sensing mechanisms will be discovered. We need to better understand the cross-talk between the microbiome and the gut, same for the skin and other surfaces. Many microbial and dietary compounds can affect the immune system, such as short-chain fatty acids. Of these, butyrate seems to play an important role. Among the many dietary components, the amino acids taurine and tryptophan, niacin or bile acids itself, or their microbial products affect the immune system. Among these, the immune sensing and signaling of short-chain fatty acids are best understood. Short-chain fatty acids are end products of microbial fermentation of mainly indigestible starch from the food we consume. In addition to acting as an energy source for the host, short-chain fatty acids induce regulatory T cells and tolerogenic dendritic cells, suppress interleukin-6 production in macrophages, induce production of luminal IgA, inhibit stem cell proliferation, and increase insulin sensitivity in muscle and liver tissue, and may have many more biological effects. Furthermore, there seems to be geographical differences in the microbiome-gene-diet interaction. Over half the world's population is raised in conditions which lead to growth stunting, typically a sign of chronic malnutrition. In addition to malnutrition, there is increasing evidence that the nutritional value of food is also influenced in part by the structure and functionality of the gut microbial community. And so, how the microbiome, diet, and genetic phenotypes interact will vary in different regions of the world. So, Professor Ramnik tried to find similarities or differences between regions that could be linked to autoimmune diseases. The diet from different parts of the world has a strong effect on microbial composition. Children from a rural village in Burkina Faso, whose diet consists mainly of cereals, legumes, and vegetables, have on the phyla level a significant enrichment in bacteroidetes and a depletion in firmicutes, unlike the European children following a modern Western diet. More specifically, the children from Burkina Faso show a unique abundance of bacteria from the genera Prevotella and Xylanibacter, which are known to metabolize cellulose and xylan and which are completely lacking in the European children. Furthermore, the early life intestinal architecture and mucosal immune system differ across different geographical regions. Environmental enteropathy, which occurs in many countries with malnutrition and growth stunting, is linked with poor sanitation. Both a poor diet and early life microbial insult, such as environmental enteropathy, seems to be linked with gut barrier dysfunction, crypt elongation, and changes in mucosal immunity, as shown in an animal model. How these populations sense and respond to dietary, microbial, and environmental antigens will be different. What are the lessons learned from Professor Ramnick's research of early life microbiome composition? Delivery mode, especially caesarean section, has a long-term effect on the microbiome. Different bacterial species have different strain similarity profiles among children. Antibiotics have a detrimental effect on gut microbiota in early life. Antibiotic kids lose microbial diversity and have more single-strain species. Their microbial communities are less stable, and antibiotic resistance genes are detected following antibiotic treatment. In the second part of his talk, Professor Ramnick discussed links between changes in gut microbiome and autoimmune diseases. Finland, Estonia, and Russia have different incidences of type 1 diabetes and allergic diseases. Finnish children have a higher incidence of type 1 diabetes and higher prevalence of allergic diseases than Russian children. In nearby Estonia, coinciding with economic development and improvement in living standards, the incidence of type 1 diabetes and allergy has been transitioning in recent decades from rates similar to those of Russian Karelia towards those of Finland. These three populations provide therefore a living laboratory to study environmental triggers of autoimmunity in a large birth cohort up to the age of 3 years. Early life microbial compositions differ between these countries. In the first year of life, bacteroidetes are more common in Finnish and Estonian infants, whereas actinobacteria are highly abundant in Russian infants. What does this mean? Are there functional differences? The lessons learnt are differences in gut bacteria and consequently in microbe-associated molecular pattern, or MAMPs, may affect immune maturation. Early life endotoxin exposure, such as LPS from E. coli in Russian children, seems to educate the immune system, leading to a decreased risk of autoimmune diseases later in life, predisposing Finnish children to autoimmune diseases. Whereas LPS derived from Bacteroidetes species in Finnish children is structurally and functionally different and less able to educate the immune system in early life. Another example is inflammatory bowel disease, including Crohn's disease or colitis ulcerosa, for which Professor Ramnick will discuss promising links to the role of gut microbiome from his research. In common, inflammatory bowel disease is very complex and require a multifaceted approach to study. Studying the microbiome of a cohort of children with new onset of Crohn's disease before treatment reveals distinct differences in microbial composition that are strongly correlated with the disease status. The changes in gut microbial composition were also linked with increased production of homocysteine and reactive oxygen species, indicating functional disturbances of the gut microbiome in IBD. Various experimental and preclinical studies point to an anti-inflammatory role of tryptophan and its microbial metabolites in IBD. Of particular interest are IA and IPA, which have anti-inflammatory effects, and IA promotes intestinal barrier function and seems to affect the innate immune system. Microbes of IBD patients have reduced ability to cleave mucins and metabolize tryptophan. However, computational analysis identified Peptostreptococcus russellii and P. anaerobius to produce the tryptophan metabolite IA and IPA. What does this mean? Further analysis show that not all Peptostreptococcus species obtain the complete FLD-AIBC gene cluster to metabolize tryptophan and produce IPA and IA. Interestingly, metagenomic analysis of stool samples of IBD patients show an association with reduced incidence of the FLD-AIBC gene cluster and occurrence of IBD. However, there are more microbial metabolites that have a positive effect on the immune system. What are the conclusions on the link between gut microbiome and health? Early life microbe-immune interactions set the stage for future susceptibility to disease. The role of early life LPS is such an example. There are distinct microbial signatures in the microbiome of IBD and type 1 diabetes patients. Susceptibility to autoimmune disease in different regions of the world may have more to do with environmental factors than genetic factors, as in Russian versus Finnish children. Microbial metabolism provides beneficial signals to the immune system, contributing to host health, as shown for the tryptophan metabolites IA and IPA. What is the outlook? Techniques to manipulate microbiotas are needed. Although we have a lot of data on the composition of different microbiotas in relation to many phenotypes, we need techniques to shift microbiota A to microbiota B. Microbiome-based therapies could be the future in treating autoimmune diseases. This would request a change from the classical thinking about changing gut function through probiotic intervention to a next generation of individual probiotic based on patient metadata and metabolic output and/or including intervention based on adequate microbial metabolites. How should we look at the microbiome moving forward? The microbiome is a complex adaptive system. It should not be thought of in isolation, since it does not exist in isolation. It is an ongoing conversation with the host. There are blurred lines when it comes to disease. Professor Ramnick suggests to move away from the single microbe hypothesis that is responsible for any disease, such as allergies or autoimmune diseases. Furthermore, we need to look at the microbiome in its context. Assigning a tag of good or bad to a bacterial function in the context of host disease is oversimplified. The microbiome is not a new trend. It is a variable in every aspect of host health which was previously dismissed. But do not oversell it. The microbiome will not be the cure to all gut diseases.